everyone, I am Afifa Naushad, and today I am going to try to explain to you a little subtopic of molecular genetics, which is DNA replication. So we will break the word DNA replication into two parts. The first is DNA and the second is replication. So first, let us have a little gist about what DNA actually is. DNA, according to the Watson-Crick model, looks something like this, which is composed of two strands which run anti-parallel to each other, which are wound around each other in a double helical fashion. It is composed of small units called nucleotides, and the nucleotide has nitrogenous bases which are connected to each other by the hydrogen bonds. Okay, coming to the next word, replication. So what does the word replication mean? Replication means to make a copy. So there is a particular pattern that DNA follows to make a copy, and that pattern, that method is called semi-conservative method. What does semi-conservative method mean? So semi-conservative method means something like this. Suppose if this is the DNA molecule, the two strands are wound around each other in a double helical fashion, as you can see here. In semi-conservative model, what happens? The two DNA strands actually separate out, something like this. And a new DNA strand is synthesized for each. And each of them undergo winding around each other to form the double helical model once again. So, in DNA replication, we are going to concentrate on the prokaryotic DNA replication because it is a more simpler model to study than eukaryotic replication. We are going to study about three main molecules in the initial steps of replication, that is DNA gyrase, DNA helicase, and single-stranded binding protein. So if we consider this as the DNA molecule, which is double helical in nature, two molecules come in, the DNA gyrase and the DNA helicase. Now the function of DNA helicase stands that it breaks the helix, as very much the name suggests. It kind of unwinds the DNA strand and makes a replication fork, something like this. Now next comes in the activity of DNA gyrase, which is to break the supercoil which gets formed due to the helicase activity. Now to demonstrate this activity, I will need the help of two filaments, something like this. If we consider this to be the DNA strand and helicase is opening the strand, something like this, as you can see here, there is formation of a supercoil, which is not allowing the DNA to be opened further. So to break this supercoil, we need DNA gyrase, which cuts and reseals the DNA molecules over here. And then the activity of the single-stranded binding protein. What they do is that they go and bind to the free strands of DNA so that they do not again recombine to form a proper DNA helix, because we will need these strands as templates to synthesize new DNA strands. This is the orientation. The DNA helix, the replication fork, the DNA gyrase, the DNA helicase, the single-strand binding proteins which are bounded on the outside of the DNA. Now next molecule that is coming in is the primase, which is going to synthesize an RNA primer. Now this primase is a DNA-dependent RNA polymerase which is going to synthesize a small strand of DNA. Now, any strand that is synthesized, be it DNA or RNA, is always synthesized from the 5' to the 3' end. Now, for this particular strand, this end is the 3' end. So, because DNA is anti-parallel, for the new strand that is going to be synthesized, this end will be the 5' end. So, the RNA primer has synthesized from this end and it's going to continue synthesizing till the end. But for this particular strand, the 5' end is on this side. So obviously, the new strand that is going to be synthesized here will lie the 3' end and the 5' end will lie towards this end. So the RNA primer is located somewhere over here. After the RNA primer comes in, the next molecule that comes into play is the DNA polymerase. Now here we are going to talk about the DNA polymerase 3 because it has the highest processability in the 5' to 3' direction. There are other two varieties of DNA polymerase as well, DNA polymerase 1 and 2. We're going to come to that later. But for now, DNA polymerase 3 is the molecule which is going to bring in the nucleotides, make a new phosphodiester bond and synthesize a new molecule. So, suppose if we begin with this end, it's going to bind over here and synthesize a new molecule of DNA, something like this. And if it sits here, it's going to also synthesize a new molecule of DNA. So this is how a next step should look like. This particular strand has started to get synthesized from the 5' end and has ended till the 3' end. And this also has started from the 5' end and is continuing to get synthesized. In the continuous, it will happen continuously that the DNA guylase and the DNA helicase, which I have removed from here, will continue to open the strands and make a wider replication fork. So we've come on to the next step. Here what will happen, suppose the two entire molecules of DNA have been separated. For simplicity we are showing it like this, but it actually does not happen like this. Another molecule that comes in here is the DNA polymerase 1. We had spoken about DNA polymerase 3, which has the highest 5', 3' synthesizing activity of DNA. Another DNA polymerase is the DNA polymerase 1, which also has that activity, but not as much as DNA polymerase 3. Now, the special function of DNA polymerase 1 is that it has RNA primer eating capacity. Means if it comes in here, it's going to catch up all the RNA primers that are there like this, and the gaps that have been created because of that is going to be filled by another type of DNA glue molecule that is called the DNA ligase. Means if the DNA ligase are coming in, what will happen, they are going to synthesize new strands of DNA which is going to seal the ends somewhat like this. So here we see that this particular strand has been synthesized in bits and pieces, which are called the Okazaki fragments. And this strand is called the lagging strand. And this particular fragment of DNA has been synthesized continuously, and this is called the leading strand. So the leading strand, lagging strand composed of Okazaki fragments. And the final step will obviously be curling or the twisting of the DNA strands to form a double helical pattern. So what we have seen is that from a single DNA strand that is comprises of, a single DNA molecule which comprises of 2 DNA strands, we get 4 DNA strands comprising, that is basically nothing but 2 DNA molecules.